Okay, good morning, everyone. I would like to start by thanking the organizers for inviting me to give this talk today. And what I'm going to talk about is about how modeling combining multiple gene alterations can help in improving the identification of drug-sensitive tumors. And in particular, I'm going to talk about the importance of recall in this kind of model. So that's how we can improve it and therefore reduce the number of false negatives that are in our models. So, as you may know, precision oncology is about finding the right treatment for each patient, and that has a component of prediction. So how do we predict which patient responds to a drug? And this here, or more like here, you will have what is a usual workflow in doing this modeling. It will start with some form of molecular profiling, generally has been DNA sequencing to detect somatic mutations in the tumor, and then there will be some level of integration between the molecular profiles and the drug response to generate a model that can predict drug outcome from the patient's genomic signature. So the goal, or how we use this model afterwards, is to predict drug response from the molecular profile of the patient we want to predict response for the treatment, right? Now, most studies so far have used one-dimensional model, which is essentially identifying which somatic mutation in the tumor is most discriminative between patients that respond or not to a particular treatment. And this has a number of limitations. First, the single gene markers has only been found for a few binomial drug cancer types. So, for instance, erlotinib treating this several forms, or this form of lung cancer. So, which means that few patients currently can benefit from this kind of models. And it's also the case that even when one is found, simple one-dimensional models tend to provide a modest prediction of drug response. So just to give an example, this is in this study, they presented data for 100 non-small lung cancer patients that were treated with erlotinib. And here you can see that only 3 of the 19 EGFR mutant patients were responsive, so that represents a precision of 16%. Furthermore, only 3, and I don't know you are reading this well, but only 3 of the 9 erlotinib responsive patients, here represented by this green circle, were correctly predicted by such single gene marker, which represents a recall of 33%. And the other 6 responsive patients were wild type for the marker, so it means that they respond by some other mechanism. And a more complex model might be able to actually capture this interaction between genes and provide a better model for these and other cases. Even though it's an FDA-approved marker, the level of predictability that it provides, at least with this data set, is 0.11 on MCC. MCC is massive correlation coefficient, and the level of random performance is 0 when the maximum is 1. So it is a hard problem, and there are a few cases where we do, where these predictors are very good, and then many where performance is much more moderate. Now, how do we generate new models to predict drug response? One limitation is that complete and curated clinical data is scarce, right? So what it means is that for many drug cancer types pairs, we lack either the responses of the patients to the drug or the genomic profiles of the tumor, most of the times it's actually the responses of the drug responses, because clinical data is very hard to integrate, to extract the right information, to put it in the right format, etc., etc. So it's much more easier to actually take a sample and do all the molecular profiling. So these actually tell us that we need preclinical data. And in some cases we'll have data at different models, perhaps for the most relevant to patient, which is actually patient data. But in many other cases, we only have perhaps patient-derived xenografts, so mice models, the response of these animal models. But in many others, we have cell lines. So what do we do when we don't have anything but cell lines? We use them, right? We at least have some kind of predictability for that case, even though we know that cell lines have some issues and limitations, and they are not so relevant for as other models to patients. So I'm going to talk now about how we compare single gene and multi-gene predictors of cell lines. And you may be aware of a study 6 years ago called the Genomics of Drug Sensitivity in Cancer, or GDSC, where they were searching for new single gene markers. And the workflow of this work was generating new data sets. So it is an example of how generating new data sets and their systematic analysis, a typical workflow or traditional workflow, where drugs are screened on a large panel of cancer cell line. A phenotypic readout is made to assess the intrinsic sensitivity of these models to the drug. Then a molecular profile of the untreated cell line is determined. And finally, a parametric statistical test is used to identify significant drug-gene association or single-gene markers. Beyond single-gene markers, we have, in collaboration with some of the authors of this study, generated multi-gene predictors, so models that combine different gene alterations. In this case, in this example, we use multitask learning. And in a subsequent study, we have been comparing directly single-gene markers with multi-gene predictors. And the question is, will combining multiple somatic mutations result in better prediction of which cancer cell lines are sensitive to a given drug? For that, what we did was to take all the initial data that was provided for each drug, extracting the single-gene markers that were detected with this data, and using exactly the same data, we were generated a machine learning model combining this mutational data. And then we were testing both approaches on a test set using featured data. That is data that was not used to generate any of the models that were tested. There was no intersection between both data sets. And this is an example of the results that you get. So you can see there is the best single gene marker that was studied for this study is the MYCN mutant cell lines are predicted to be sensitive to this drug, which is a checkpoint inhibitor. Then you can see that the sensitivity threshold will tell us what is sensitive or responsive. Is in red, so it is the median IC50 or half maximal concentration of the drug on the training set, and then we can calculate a measure that actually captures both types of error, false positives and false negatives. And what we observe on the training set, it has quite moderated performance. In the test set, this is slightly worse with 0.07. Now, to generate these multi-gene markers, multi-gene models, we are using machine learning with built-in feature selection. We used a fairly standard approach, which is a gold standard in terms of using this data set, because it is appropriate for high-dimensional data. So there are algorithms that ignore features that are non-discriminative, so they form an internal form of feature selection. Random forest is one of these. And essentially what it is, random forest, is an ensemble of diverse trees. And each of these trees is formed in such a way that for each of the terminal nodes you have partially similar tumors that have similar response. So you use that as a model. Now, when you compare both the single gene marker with the multi-gene marker on the same test set for this drug, what we see is that the multi-gene model obtains 3 times better MCC than the single gene marker. And I highlight again that this is actually a performance level that tells us that it's a hard problem. But what is interesting is that actually it's considered to be very good, because the precision, you know, it is 78%, which what it means here, the precision is how is the hit rate, so how much of the actual predictions that we do are actually correct. It's a 78%, but the problem is that the recall is quite low. So what it means is that while there are lots of false negatives in this case, so that is cell lines that are predicted to be resistant but are actually sensitive. When you are combining, considering multiple mutations other than the one for the single gene marker, you get many more of these cell lines correctly predicted. And when we look at all the 127 drugs, we observe with MCC, we observe a large variability. 55% of the drugs obtain better MCC when using the multi-gene model. And 9 of the 14 cytotoxic drugs that you can see here in red had better MCC by combining multiple genes by a random forest. We look at precision, so precision capturing the number of false positives. In this context, that would be the proportion of cell lines predicted sensitive that are actually sensitive. We also observe a large variability and a slightly worse performance for the multi-gene models. But when we look at recall, which is essentially capturing the number of false negatives, the picture completely changes. It's essentially 93% of the drugs obtain better recall when using multi-gene models. Now, in a more recent study, we have been looking at patient-derived xenograft, PDXs. And so this data come from this study from Novartis, the PDX encyclopedia from Novartis. And essentially what they did was to take about 1,000 PDXs. They tested, they treated the PDXs with 60 treatments. And for a fraction of them, like about 40%, they determine 3 tumor profiles: mutations, copy number, and gene expression. The 2 types. So the thing with large data set is that when you look about which part of that is useful for the question that you want to answer, then you're looking at the intersection of the data, and then data sets are not as large as they can see at the beginning. So we had around 40 to 50 PDXs for 2 types of 2 cancer types in the data. So that was breast cancer and colorectal cancer. For each type, it was treated with 13 drug therapies. And what we did is modifying random forest to calculate the optimal model complexity in a way that we were using for the modeling only the most predictive models. And let us tell the model, the data itself, where you do the cutoff in terms of which are the most predictive features. So this is just a slide to mention something that you probably all know, that is the high dimensionality of data is challenging. In many cases, you may have things like 40 PDXs, 40 data instances, but each of these is profiled with tens of thousands of features. So this poses a challenge for supervised learning. And here you can define dimensionality like the number of features divided by the number of data that you have. In this toy example, we have model 1. It's built on high-dimensional data, therefore in green is too complex. Overfit the data. New data is probably going to be suboptimally predict. Whereas you look at model 2, it has the right complexity, is more likely to generalize well, right? And this right complexity is the challenge of the algorithm is trying to ignore or exclude as many relevant features as possible. That's a way to try to strike this balance. In terms of the validation, what we have done is fairly standard, so it's cross-validation to measure performance. You're probably familiar with this, but essentially it's a stratified fold cross-validation. Every PDX is exactly once in a test fold, so there is one predicted class per PDX. Any PDX has its actual class, so we can just get which are the number of true positive, false negatives, etc., and then summarize this confusion matrix into a number, which is the NCC. In terms of what this optimal model complexity does, the motivation is that once you have this problem, either you have more data, which is normally not available, or you retain the most informative feature as a way to handle this high dimensionality. And OMC, what is a strategy for the data-driven identification of the subset of these features. And I can go quickly on what it actually does, but essentially when you're looking at the training falls, you are calculating a p-value for each feature across between each feature and the response to the drug across all the data instances. Then you rank the features to see which are the most relevant for the problem. Consider in the model selection step to n divided by 2 nested feature subset. So it means that yn divided by 2 is just to ensure that we have at least 2 data instances per feature. And among this model, we select the model that has the highest inner cross-validation. There is a second cross-validation going on here. And then the selected model is trained on all this data and tested on the outer fold. So these are just 2 examples of the best models that we got with this data. So for binimetinib, using gene expression and on breast cancer, so when treating breast cancer, the model selected 14 features. What you can see here is each of these lines is a PDX, right? Is how the tumor of the PDX growth with time, right? And you can see that the red are predictive resistant, the blue are predictive sensitive, and the false predictions are in dashed lines, where true predictions are solid lines. So here you can see that we are getting an MCC of 0.57 in this case, and in the other case, best case for the colorectal cancer, cetuximab on colorectal cancer using single nucleotide variants, so mutations on the genes. This model selected the top 4 features and is also a good model for this data set. But importantly, I'm coming back to the recall of these models, right? When we are comparing single gene markers with multi-gene markers on this data set too, you can see that most of the treatments have, or the models for each treatment, so these are the 13 models here and 13 models, 1 for each treatment, they have higher recall, so it means they are able to identify a higher proportion of sensitive tumors, right? The possibility of predicting which are the sensitive tumors are limited in terms of the single gene markers, because single gene markers are a mutation, and mutations generally have a low prevalence, right? So if you combine mutations, you can get a better prediction, a better recall, so identifying more of the sensitive tumor, which I think is a very important feature. So just to sum up, I would like to say that what we have seen is that multi-gene models often are more predictive than single-gene markers, and we have shown this in vitro with pan-cancer data and in vivo with cancer-specific data. Multi-gene models generally have higher recall, and with few exceptions, single-gene markers have low recall, so responsive tumors without the marker are missed. And consequently, combining the mutational status of multiple genes via machine learning should be always considered. It might not be the best model for that particular treatment, but in some treatments it will be the best model, so we are missing opportunities by not using them. And this variant or random forest has this advantage. In some cases it's better than when we are using all features with random forest, but it has also the advantages that give us a gene list of which are the genes, so how many genes are actually the most predictive for a particular case, which is beneficial for clinical implementation and interpretability feature of the data set. And the methodology can also be applied to other tumor profiles like microRNA, DNA methylation, etc. So I just would like to finish by acknowledging the help of different members of the groups and current funders, and to all of you for your attention. Thank you.
Thank you very much, Pedro. Any questions? I can just a bit worried about asking questions. The fact that you find that multi-genes is more important than single gene, is it because you believe it reflects more the heterogeneity of the tumor and therefore the sensitivity to the drug, or is it because you're bringing more data to your model?
I think it's the first one, is the intertumoral heterogeneity. The fact that there are a proportion of the tumors they respond by a particular mechanism, where there are other tumors that respond by another mechanism. The moment we go with a hypothesis embodied by the single gene marker, we are choosing one of these hypotheses, whereas in a model we can actually integrate different hypotheses in the same model.
No, there's going to be a lot of discussions at coffee break.
I hope so. Thank you.
Okay, so thank you very much, Pedro.